actually didn't need to post this, or I did, and then I changed my mind. So if you downloaded this PowerPoint early this morning, you probably saw this clicker question on there. It's just meant to be kind of a joke, because it's something I saw last night, and it's messing with my mind. So what color is this?
Oh, yeah.
All of So who sees it as white and gold? Blue and black. Whoa, that's weird. So last time, like I asked like 10 people yesterday, and everyone said white and gold. But this, I mean, it looks blue and black to me, but I was like the only one yesterday. And I still can't wrap my head around why that is. Did it switch for anybody? Like if anyone looked at it yesterday, yeah, it switched for me.
I think it's because I'm closer up.
But now it's back to blue and black. I thought it would change again. So the only reason that I show you, well, not the only reason, one, it's kind of goofy when you start the lecture. And it would be really awesome if I had a good way to transition this into transcription regulation, but it doesn't as far as I know relate anyway. So it's just for fun. And now we'll go on with the real lecture. So like I said, we're going to talk some bacterial transcription regulation. Basically, we're going to try to hit all of these topics today. You guys talked about eukaryotic transcription last time, and more of not just transcription regulation, but some like basically everything. And you're going to talk more about eukaryotes in the next lecture, which is kind of the backwards way that we usually present this stuff, and all textbooks do it differently because prokaryotes are, let's just say bacterial transcription regulation comes first because it's the simplified version, and then you get into the eukaryotes, and those are crazy and do all kinds of weird stuff, and it's not nearly as well understood. So now you guys will get the easier stuff, and then maybe the eukaryotic stuff will start to make a little more sense. So we're going to talk basically first about some negative regulations, then we're going to hit some positive regulation, lots of examples of positive regulation today. Lambda repressor, we like to use viruses as examples, especially Ken, if you guys have noticed, he uses viruses whenever he can, because that's what we do. So this is a really cool example of some regulation in a bacteriophage, which still qualifies as bacteria, I think. We'll hit the lac operon pretty hard. You guys have probably heard this every time you've taken any kind of class that deals with molecular biology. So we'll cover it again, because we like to cover it over and over, and you get to relearn that. We'll go over some practical applications, and then if we have time, I hope we can get to the rest of this stuff. So as we go, just ask questions. I think we're going to have plenty of time, so if anything doesn't make sense, which it probably won't, just ask me, and hopefully we can talk it out. So again, transcription regulation. You guys know from your last few lectures that you can basically regulate the gene expression, so going from DNA to the active protein, you can regulate that process at almost any one of these steps, right? You can hit it at transcription, you can hit it after transcription, so once you have the RNA, you can regulate if it becomes mRNA, you can regulate if that mRNA makes it into the cytosol from the nucleus. Is that going to be a problem for bacteria? Can you regulate that, the RNA traveling from the nucleus to the cytosol in bacteria? Yeah. Can you?
No.
They don't have a nucleus, right? So that's not a problem in bacteria, so they don't have that control step. Bacteria, just like eukaryotes, can regulate translation, so you can regulate if that RNA is translated into a protein. And then once the protein's made, bacteria, just like eukaryotes, can do those translational, post-translational modifications like phosphorylation, ubiquitination, those kinds of things that'll influence the activity of the protein. So which one of these steps is the fastest way? If you wanted to regulate gene expression, the fastest, which one would you hit? You'd hit it here at the protein, right? And once you have the protein made, it's easiest to turn it on and off, right? And Ken talked mostly about that last time, where phosphorylation is a very common way to turn a protein on or off. You have those enzymes called kinases that are floating around the cell, and they can either pop a phosphate group onto a protein to flip it on or off, and then something else can take that phosphate group right back off and do the opposite. So you can flip the protein on, flip it off without having to do all of these steps before that, right? So you've got the protein there ready to go. You can turn it on and off, right? So if you wanted to respond very, very, very quickly to something, that's how you would do it, right? We're going to talk about transcription regulation. So the reason that transcription regulation is very, very highly regulated in both eukaryotes and prokaryotes is one, why go through all these steps if you don't need to? It's a lot of energy to basically transcribe a gene, take that RNA, and then translate it, right? So it's very energetically costly for the cell to do that. So if you don't need to make a fast response to something, if you have the time to maybe wait and then transcribe the genes and then make the protein, it's probably more efficient for the cell to just shut down transcription until you absolutely need to go through all those steps. It's also the easiest step. We can argue that it's the easiest step, but once you, basically the reason I think it's one of the easiest steps is There's only typically, in a bacterial genome, there's typically one gene, right? And that one gene has one promoter, so you only have to regulate that one single promoter, right? So if you block that promoter or activate that promoter, you get to pick if that gene's on or off, right? You just have to block or activate one thing. If you let that gene get transcribed into RNA, then you have tons of RNAs floating around, right? And you have to then block each one of those RNAs. And then if you let it go to the protein and you want to regulate protein, you have to then block every one of those proteins, right? So there's a lot more of these things floating around if you let it get past the transcription step, yeah.
So when you say there's only one gene per bacterial genome, you mean
One gene
per
per, like,
RNA?
What's that?
You mean per RNA?
No, so the gene is going to transcribe a single RNA, right? So one gene codes for one RNA, but that gene's going to make a ton of RNAs in one go, right? Once you turn the lac operon on today, it's just going to transcribe that thing like crazy. So you're going to have that activation, and then you get just a cascade, like a huge production of tons of RNAs from that one gene. Does that make sense?
Yep.
Yeah, so whenever you transcribe a gene, you're not just making a single copy of RNA, you're making lots and lots and lots of copies of that RNA because you want to get a lot of that signal out to the cell. And the cell can regulate how much of the copy to make, and some are better than others. You guys talked about promoter strength, right? Stronger promoters are going to lead to more active transcription, that kind of thing. If you have a weak promoter, you're probably not going to make very much RNA. But that's getting off topic. So again, it's energetically efficient to stop it at transcription. It's easiest because it's only one spot to regulate. Why not though? Why would cells not want to regulate transcription? And we already kind of talked about that, right? You want to be able to respond to something very fast. So if you want to be able to respond to something fast, you probably want to have that protein already there and you can just flip it on or off. Or maybe have that RNA just ready to go, ready to get translated. Cool. So is that good? You guys saw this figure. It's just a diagram of all the spots to we're again focusing there today. So you guys talked about DNA binding drift, right? DNA binding proteins you've already hit. Okay, so this is just review then. So transcription factors are what we're going to focus on mostly today. So when we talk about repressors and we talk about activators, they're just called transcription factors. Your book calls them transcription regulators. It's all the same thing. They're all DNA binding proteins, so they've got to be able to recognize a sequence of DNA and bind to it. And they've got to usually recognize a sequence-specific piece of the DNA. You don't want them just binding anything, right? Because then it's not going to be able to regulate anything at all. Your book calls those DNA sequences that the transcription factors bind to, they call them cis regulatory elements because they call them cis because they're on the chromosome of the gene that they're going to regulate, right? They're not on a separate chromosome. And that's always going to be the case for bacteria because bacteria only have one chromosome, right? It's usually just a single circular chromosome. So all of the DNA sequences that are regulated by these promoters or by these transcription factors are all cis regulatory elements. And then your book kind of, I think it uses this, but other books have used this in the past, and this is what I was taught. They're called operators, so I'll probably use that word a lot. So operator also just means the DNA sequence that's bound by the transcription factor. And then this is probably good to know. So helix turn helix motifs in the transcription factors, that's one of the most one of the most common DNA recognition motifs used by these transcription factors, yeah.
So just to clarify, operators is a category that includes pretty much all cis regulatory and trans regulatory elements?
So operators are you can just think of them as the DNA sequence that is recognized by a transcription factor or a regulatory protein, something like that. So it's the DNA sequence itself, yeah.
But it's non-specific. It's not specific.
It is specific for a certain transcription factor. So one transcription factor is generally going to recognize one operator sequence or a closely related set of operator sequences.
Right, but what I mean is like operators, it's a non-specific term for
Yeah.
pretty much any kind of
Yeah,
sequence.
exactly. Yeah.
Okay.
So operators encompasses any kind of DNA sequence that's recognized by a transcription factor.
Okay.
So the lac operon's going to have operators in it. The arabinose operon's going to have operators. Those operators are going to be different from each other, but they're going to still be called operators, if that makes sense. So you guys talked about the structure of DNA, and you know it has a minor groove, the narrow part of the double helix, and then it has the major groove, which is the wider part. So the major groove is typically where these transcription factors are going to bind. I'd say like 95 to 99% of them are going to bind in there. So why is that? What do you guys remember from that? Why would that be the place to bind?
Specificity is easily recognized.
So what do you mean by that? It's easy. Why can't you recognize does the minor groove not have specificity in it?
Well, I thought that the specific bases were made imminent based on the molecule.
Yeah, so both the major and the minor grooves are going to present bonding. They're going to present hydrogen bond donors and acceptors, right? They both have patterns that can be recognized by a DNA binding protein. So you're right. Just the major groove is bigger. So it's easier for a transcription factor to fit in there, to fit that helix turn helix in there. So that's why most of them are going to bind in there. This is just a figure showing that in addition to the information coded by hydrogen bonding between the bases, the AT and the GC, you know how they know how to hydrogen bond to each other, they also have additional hydrogen bond groups that stick out in these major and minor groups that are what's recognized by the DNA binding protein. So the DNA binding protein is going to use its helix turn helix. So here's that helix. It's going to insert it into this, in this case, the major group, and it's going to recognize some of these contacts. And each base is going to present different contacts that different amino acid side chains can then recognize. So that's how you get the specificity. So you say an operator sequence binds a or a transcription factor. The operator sequence presents that information through these grooves. Typically the major group, it presents hydrogen bond donors, hydrogen bond acceptors, hydrophobic interactions that this helix here can recognize specifically. Whereas other helixes from other transcription factors can't recognize. Okay, and that's the lambda repressor, and we're going to come back to that in a second. So you have basically two categories of.
Ken said that alpha helices are the perfect size to fit in major grooves. Do beta sheets fit in minor grooves?
Probably, but they're not more perfect to fit in there. They're still too big to fit in there.
Okay.
There's always exceptions, right?
Yeah.
Typically, what's going to bind in the minor group, and I bet Ken talked about this, is non-specific proteins. I forget, I think TDP, the saddle protein from eukaryote transcription, one of the transcription factors, it binds in the minor group. Non-specific, well, it does make some specific contacts, but actually, yeah, it does, absolutely, because it binds to TATA binding protein. So it binds in the minor groove, and that has to happen because it bends the DNA. So proteins that bind in this minor groove typically are going to cause bends in the DNA. And what does that bending do for transcription in eukaryotes? It's really, really helpful. Why? You guys remember?
Platform for the
So TBP creates the platform. What does it do to the DNA? When you bend DNA, you distort it. And then what happens further downstream where you want to start transcription? Do you remember? Maybe he didn't talk about it. So did he talk about it opening up, like the DNA, it's easier to melt? So the binding of TDP in here bends it in such a way that downstream it's easier to melt apart so that the RNA polymerase can come in and then start transcription. That, again, doesn't really matter for today, but that's just an example of what happens in the minor groove of binding. Again, so things that bind in the minor groove are typically going to cause architectural. So things that bend DNA are typically going to bind there. Transcription factors mostly bind in the major group. And again, always exceptions, and we always talk about the exceptions because they're what's interesting for some reason, and they're tricky for the test, I guess. All right, so negative and positive regulation are the two flavors of gene regulation in bacteria. And typically, you're going to have both of them working together to control the expression of a gene. It's not rare, but it's less rare, I think, to see one or the other have repression by having negative regulation only or positive regulation. So when we talk negative regulation, all we mean is there's a repressor protein, so a protein that's going to prevent transcription. So the repressor, which is shown in green here, and this is your promoter. I think this is probably the -10 element, and this is the -35 element. So the repressor in this case is binding smack dab in the middle of your promoter, and it's basically going to prevent transcription just by blocking RNA polymerase. It's going to bind very tightly to this DNA, and if it's bound tightly to those elements, the RNA polymerase can't access the promoter, and if it can't access the promoter, it can't start transcription, right? So in this case, you've got repression. You have your gene off. In order to relieve repression, something has to happen to cause this bound repressor, which is bound to its operator sequence. It has to fall off of it. And in this case, it's going to have the addition of a ligand. So something that's going to bind to the repressor. It's going to cause a conformational change in the repressor protein, which causes it to release from the DNA. So it's really tightly bound until something hits it, like a ligand, and we'll talk about one example in a minute. And then that just releases it. So do you guys remember what kind of protein that's called? Something that's influenced at a secondary binding site that affects the binding site of another site.
Allosteric regulation.
Allosteric, yeah. So all these proteins, and this is another common theme, most of these are going to be allosteric proteins. So the ligand binds and releases it from binding to the DNA. So in this case, you have, so this is kind of confusing here. So it can be activated or repressed by a ligand. So we're talking about a repressor that can itself be activated or repressed, right? So in this case, the repressor is being deactivated. Let's think of it that way. So the ligand binds and deactivates the repressor. In this case, that repressor is being activated by the ligand. So the ligand binds to the repressor, it causes a conformational change that now allows it to bind to DNA. So it can be either way, but they're both versions of repression, right? And we'll talk about both examples today. So the tryptophan operon is one of the most canonical examples of this. It's in every textbook, and it's really, really simple. Again, it's an allosteric protein, the repressor itself, and it binds basically right in the middle of that promoter. So if it's bound, it blocks RNA polymerase from binding, and you can't get any transcription happening. So if you have low tryptophan, so here's the operon. So remember, bacteria have their genes all arrayed in operon, not all of them. But for energy metabolism pathways, if you want to metabolize a sugar or a certain type of thing, or you need to make a certain chemical, you're going to have a lot of those genes that need to do that all in order under the control of a single promoter, right? It seems very efficient. You can get them all made in one go. Do eukaryotes do this? No. So this is pretty much just bacteria and archaea. So it is prokaryotic. So in this case, we want to make tryptophan or we don't want to make tryptophan. So if we don't have any tryptophan in the cell, we're going to want to make it, right? So that repressor is going to be inactive. It's not going to be bound to its operator sequence, and transcription is going to be able to proceed just fine. The RNA polymerase binds by itself, doesn't need any help. It's got a strong promoter. As long as it's able to access the promoter, you get transcription. You'll get all the genes you need to make tryptophan. However, as tryptophan levels begin to rise, because you've now turned this operon on and you're starting to make your own tryptophan, or if you add tryptophan to the cells, if you start giving them tryptophan, tryptophan is actually the molecule that's going to cause the repressor to bind back to that operator sequence and is going to turn off transcription, which is kind of handy. It's negative regulation, right? Once you have enough of the thing that you want to make, you want to be able to shut that operon off. So tryptophan is the thing that's going to be the ligand in this case. It's going to bind to the repressor, activate it, clamps down on the promoter, and you get no transcription. So it's a very clean mechanism, right? And then once tryptophan's levels start to run out, the cell starts to use all its tryptophan, it becomes dissociated, and you can turn it right back on. So on, off. As more tryptophan comes, it turns off. As tryptophan rises, it turns back on. So it just kind of fluctuates depending on if there's tryptophan or not. So that's probably the most straightforward one that we're going to talk about. Positive regulation is the opposite. So instead of repression, you're going to have activation. So you're going to help turn the gene on. So remember, some promoters just suck. They're intrinsically weak, meaning RNA polymerase can't bind by itself, or it can't bind very well. And if it can't bind very well, you're not going to get very good transcription, right? So that's one way a cell can control whether or not it wants to even have that gene being transcribed, right? You can just make the promoter be crappy, right? If you have a crappy promoter, you're not going to get good transcription. And the only way you would get good transcription is if you activated it. So the activators promote transcription. It's basically the same kind of thing. A lot of these guys are allosteric. So it's not going to be, in this case, it's going to be activated without the ligand bound. So when the ligand is not there, the activator is able to bind to its operator sequence, and it's going to activate RNA polymerase, and the gene's going to be on. The important difference between this and repression is repression is basically physically blocking, and it binds basically in the interior of the promoter. Remember that operator sequence? If we can go back. The operator sequence is in the middle of the promoter here. For activators, it's typically adjacent to it, so it's not going to be in the middle. And that makes sense, right? You don't want to have your activator binding inside of the promoter because it's just going to block RNA polymerase. They're typically adjacent. And then the other thing that's a good take-home from this is that the activators usually make but they make interactions with the RNA polymerase. So RNA polymerase basically makes some kind of bond, not a permanent covalent bond, but some kind of interaction that helps stabilize the RNA polymerase on this promoter sequence. Because remember, it's a weak promoter generally. RNA polymerase can't bind on its own. It needs the help. So this basically binds really well to its operator sequence. RNA polymerase binds really well to it, so it kind of positions RNA polymerase in the right spot, and then RNA polymerase can go and do its thing. Again, you have the addition of a ligand in this case, whatever that ligand may be. It's going to bind to your activator, and it's going to remove activation. So binding of it causes conformational change in the activator protein, and it releases from the DNA. This is the opposite scenario. The activator, or did I just say that backwards? No. So in this case, you have your ligand bound to the activator. It needs to be bound in order to get activation, so you have to have it. And then once the ligand is not there, you basically lose activation. So again, just like repression, you have two different flavors of it. The ligand can either activate or deactivate the activator, or the ligand can either deactivate or activate the repressor. Yeah?
So the activator is on the left side, and then
This is the activator in green, yeah, and this is your RNA polymerase here. Yeah, so the repressor is typically, not always, you have exceptions, but typically the repressor is going to bind somewhere in here. It's going to bind within the promoter typically and block the polymerase. Activators bind adjacent to the promoter typically. And they're typically going to be, what is this, upstream or downstream of the promoter?
Upstream.
Yeah, upstream of the promoter. If it was downstream of the promoter, why would that be a problem? There are examples, again, there's always stupid examples, but why would it be more rare to have an activator bind downstream of the promoter?
Block the polymerase.
Yeah, RNA polymerase could bind, but then it's going to run right into that activator. And then sometimes the activator can bind, and then once RNA polymerase is there, the activator releases, and then the polymerase can go. So it's, as long as the activator is like there, it helps it bind, but it's kind of stuck until the activator goes. So again, always, always, always weird examples. Cool. So we're going to talk a lot about this in a minute, and this is a good example of an activator. So we call this the lambda repressor in quotes because it's both a repressor and an activator. And that's also fairly common, I think, in bacteria, having these things actually have a dual role. So the way this one works is you have, basically you have two genes. You have this called the C1, which encodes your repressor, and you have Pro, which encodes a different repressor that you don't really necessarily need. So if you want the cell to be lysogenic, if you want your virus to be lysogenic, so lambda is a phage of bacteria, if you want the phage to be lysogenic, meaning that it just kind of integrates itself into the genome of the cell and the cell just goes on living, it replicates its DNA, it replicates the viral DNA, and nothing really bad happens. The virus hasn't killed the cell. As soon as the cell gets hit with some kind of stress, like UV irradiation or like nutrient deficiency, something bad happens to the cell and the cell starts to think, oh, I'm going to die. That triggers lambda, the phage, to go into what's called the lytic life phase, where it then starts rapidly producing itself and then it explodes out of the cell and goes to find a new home. And that makes sense, right? If your home's going to get destroyed, if the cell that you're living in is going to die, you want to be able to evacuate pretty quick, right? And it's basically controlled by these two genes, C1 and Pro, yeah, which are both different repressors. Sure. Yeah, sorry. So C1 is going to encode this thing called a lambda repressor. And what we're talking about now is we want to talk about it being in the lysogenic phase. So the phase where it's not making tons of virus and not killing the cell, right? So C1 is normally just transcribed. The promoter binds the host RNA polymerase transcribes lots and lots of this repressor transcript. The repressor gets made, translated, and then that repressor actually in turn binds here to this operator, which is called number 1 in this case. And what you see by binding to this operator, it prevents expression of this protein, CRO, so it's repressing the expression of CRO. CRO is a different protein that activates the lytic life phase, right? So if you want your virus to not go lytic, you want to block expression of CRO. So by expressing lambda repressor, you're repressing CRO. And in turn, what's even better about this for this gene, not for CRO, but it's good for the cell because it's going to keep the cell alive, is it also activates expression of its own. So once you get a little of this made, it starts binding here, starts repressing CRO, and then it starts to activate itself. So you get even more of it made, right? So then you get even better repression here and even better activation. But what you see here is there's 3 operators. Eventually, this level of C1 repressor, the lambda repressor, builds up to such high levels that they start to bind all of these, right? And once it's down to all 3 operators, it's actually going to repress both of these genes, and then everything's basically off. And then, this isn't so important, but induction, let's say you blast those cells with UV light, it causes this chemical or this repressor to basically break in half. You see how it's basically a dimer here. Each of these little balls is a dimer and they're connected to each other. It causes them to basically get cleaved, and once they're cleaved, they can't do their repression anymore. So once you relieve repression of CRO, CRO is able to start getting transcribed and translated, and then it starts to go into the lytic phase, right? Because then CRO can activate a whole cascade of genes that lead to the lytic phase, yeah.
And CRO is also a transcription protein, right? A binding protein.
Yeah, so it binds, and it binds to the same operator. So both of these are expressed in the same area, and they both bind to the same sequences, and it just depends which one happens to be more abundant at the time, and then that's going to lead to either lysogenic or it's going to lead to lytic. It's really cool, yeah.
So, and when you say, going back just a little bit, when you say that eventually everything gets turned off, that means that the lambda repressor eventually binds or represses its own
Yep.
the gene which encodes
For
itself.
Yeah, so everything, both of these will get turned off, and it actually folds up on itself and makes a loop, because there's actually 3 operators further down here that also bind it, and then these little heads on this side bind those, and you get a nice, it's very tight, compact little loop. And then, so what happens, I mean, protein concentrations just naturally decrease over time. So this level of this repressor is going to decrease over time, and that's just normal. So what do you think happens in that case? Are you going to get lysolytic phase to go on? Yeah, right? So as it starts to release, it binds this one best, this one second best, and this one third best. So that's another cool thing. So it's going to release from this one first, and once it releases from this, RNA polymerase can hop back on, and it can start to make more C1. So as C1 levels drop, you can make more. As C1 levels drop, you can make more. And the only way you're going to get rid of C1 completely is if you do some kind of stress. And then once they're completely gone, CRO basically just hops on, takes over, and you just get expression of CRO, which then causes, again, a whole bunch of other viral lytic genes that go on, and then the cell's basically screwed.
Out of curiosity, what else could the stress signal be? So you mentioned UV radiation, but what about nutrient deficiency?
Yeah, that's another one. It's so anything that triggers the cell's SOS response, which is a whole cascade of cellular protein that tells the cell we're in trouble, and it triggers, I don't know exactly all of them, but I know that the SOS response triggers something called LEX-A, I think, which is a protease, and that's the protease that comes in here and cleaves this. And it's needed if this protein mimics a cellular protein that looks almost identical to this, that does something else involved in the SOS response, and that cleavage is crucial to the cell's SOS response. It has to happen. And because this mimics that substrate for that LEX-A protease, it tricks the cell into basically killing itself. By cleaving this, it's over. The cell's basically triggered the living lifestyle. So yes, all viruses are pretty evil. They're awesome. It's the core bacteria. And again, CAP is similar. So CAP acts as an activator, and in some cases it will also act as a repressor. So on to the lac operon. So the lac operon is, again, a bunch of genes, 3 in this case, that you metabolize lactose, the disaccharide lactose. So the cell doesn't want to use lactose if it can avoid it. If you have glucose and lactose together with the cell's going to use all the glucose before it switches to lactose, just because it's more energetically efficient for the cell to use glucose. Even though lactose has glucose in it, it still doesn't want to use it because it still has the spin energy to break the bond and separate that into the glucose and the lactose sugars. So again, here's the lac operon. You have LAC-Z, which is basically the enzyme that causes the cleavage of that disaccharide. So it's going to split lactose into glucose and galactose. LAC-Y is a permease. It's just going to go into the cell membrane. It's going to allow the cell to transport in more lactose. So it allows you to bring in lactose to use it, which makes sense. And then LAC-A is a transacetylase, and it's not totally clear why that's needed. You can knock it out, and it doesn't seem to make a difference. So it does something with acetyl coenzyme A, and it's, again, there's argument, which is kind of interesting because this thing's been known for 40 years. So it's weird that, I guess no one cares because it's not interesting. If it's not interesting, no one's going to waste their time working on it. So again, it's used to break down lactose. So there's basically 4 different scenarios, and you should know all 4 of them, and you should know all of the operators, and you should basically just know how this thing works pretty well. He's going to probably ask, if he asks you any question, it's probably not going to be something in like normal lac operon. It's going to be like, you have a lac operon with this mutant. What happens if you put it in high glucose and high lactose, something like that. So you got to know this, and then you got to know how it's going to respond to different changes. So let's walk through it. So here's basically your regulatory region up here. You have this thing called the LAC-I, which is the repressor. You have CAP binding sites. So CAP is going to be an activator protein. So you have a repressor protein. You have an activator protein binding site. CAP is made somewhere else in the genome. Just pretend like it's always there. But it can't bind unless certain conditions are met. Here's the promoter for the lac operon itself. Okay. So how does this work? So you have, basically, let's say you've got the cell, and let's start here. This is probably the best place to start. You have high glucose, and lactose is unavailable. So if there's no lactose, you don't want to be metabolizing, you don't want to be making the genes to metabolize lactose, right? It's a waste of energy to make those enzymes. So what's going to happen is you've got your repressor here, which is constitutively active, meaning it's just always on. You're always going to have repressor in the cell. So if the repressor is available, it's going to bind to its repressor site. In the absence of lactose, and high glucose. And it's basically, it's good to think of the repressor and the activator responding to two different signals that are completely independent of each other. As long as lactose is present, as long as the lactose is absent, the repressor will bind to its site. That's guaranteed. No lactose, you've got the repressor there. And if you've got the repressor there, nothing's going to happen, right? You're not going to get activation, or you're not going to get anything happening with that operon. You're not going to get any of those genes expressed. So again, the repressor basically responds only to lactose. So what happens now if you start to, again, you have no lactose, and then you lose glucose. So the cell starts using all its glucose, and glucose supplies start to dwindle, right? But you still don't have any lactose around. Like I said just before, you still have repression because that lactose repressor, that lac repressor, always binds to its operator if there's no lactose around. What you see here now is this thing called CAMP, and it should just say CAP. So it's CAP protein. So if there's no glucose around, that causes this molecule, cyclic AMP levels, that's going to rise. So glucose levels and cyclic AMP levels are inversely related. So as glucose dwindles, cyclic AMP rises, and cyclic AMP acts as like a signal to the cell that, uh-oh, we're running out of glucose. We need to start activating some other metabolic pathways to start using some other energy sources, like lac operon. But again, if there's no lactose available, you still don't want to turn that on, right? So in this case, you've got your activator bound, but it can't activate because you've got the repressor there still, right? So the only way to get rid of this repressor is you've got to have lactose available. If you have lactose available, lactose basically binds to this repressor. And it's actually not lactose, it's called allolactose. So as soon as lactose makes it into the cell, there's some metabolism of it that occurs through other pathways or secondary reactions, and you get like it almost looks exactly like lactose, it's just got an inverted bond. So it's not that important. Just think of it as like a very close cousin to lactose. But it binds to the repressor, and the repressor gets released. So if you have lactose available, lactose acts as the ligand that releases repression, just like we talked about on that first slide. Lactose causes a conformational change in the lac repressor. If you get that conformational change, it can't bind to the DNA anymore. If it can't bind to the DNA anymore, you're going to get strongly expressed genes here because you've got the CAP protein to activate it, right? Why do you have the CAP protein on here in this? What makes the CAP protein bind? Low glucose, which is, yeah, exactly, cyclic AMP. So low glucose means you have lots of cyclic AMP. Cyclic AMP activates CAP. CAP activates transcription, right? So CAP responds specifically to glucose levels. So as glucose levels dwindle, CAP is going to get activated by cyclic AMP. It's going to bind to its site, and it's going to help RNA polymerase, right? Repressor responds only to lactose, right? So as lactose levels decrease, or as lactose levels increase, I should say, it's going to bind your repressor, and your repressor is going to leave. So again, if you have lactose unavailable, this thing's always going to be off, unless you have a weird mutation. But it's always off. If there's no lactose, you get nothing really, because you don't want, there's no reason, right? Okay. So the last situation, and I talked about this, I said if you have lots of glucose and you have lots of lactose, if the cell's in both of them, it's going to still use the glucose instead of the lactose. So how does it know? If there's lactose available, you can't get repression, right? So if it's not repressed, then it should make sense that it might be on, right? But it's actually not going to be on, and we call it, you can think of it as off, but it's actually leaky. So if there's no repressor, RNA polymerase could in theory bind, but there's no activator in this case, right? Yeah, there's no cyclic AMP because glucose is high, right? So if glucose is really high, cyclic AMP is really low. It's no cyclic AMP, no activation by CAP. So why do you think it's leaky? Why do you get any expression there at all if it's not activated?
Because they're still binding even though it's weak.
Yeah, exactly. You have a weak promoter here, but RNA polymerase can still bind somewhat, so you're going to get a little bit of expression of these genes. So it's not going to be turned off completely. Like in these cases, whenever you have the repressor, you can basically, it's off. It's basically gone. Cool. And CAP is also active elsewhere in the cell. So it's not, there's this CAP binding site, and if we're probably not going to get to it, but the arabinose operon is also influenced by CAP. So lots of genes that are involved in different metabolic pathways are also going to be activated or deactivated by CAP. And that makes sense, right? Because CAP responds to what? Glucose levels, right? So as glucose levels drop, cyclic AMP rises, CAP is influenced by cyclic AMP, and that tells the cell about energy, right? So CAP is basically a good molecule for sensing how the cell's doing energetically, right? So it's going to be also active not just in the lac operon, but in other places. Yeah, I think there's like 100 other genes.
Is the allolactose, the molecule that's similar to it, is that associated with the cell, or is it just
Is that associated with the what?
with the cyclic AMP?
No, so allolactose and cyclic AMP are two separate. So cyclic AMP is going to just bind, it's going to bind to the activator CAP. Allolactose is going to bind to the repressor.
Right, but is allolactose just floating around all the time?
No. As long as so if lactose is in the environment, some lactose makes it into the cell, and some of that lactose is in an alternate form called allolactose. It's just I can't remember the word. It's a chemistry word for like a mirror image of something like that. It's basically the two same sugars. They're just linked by different sugar bond, and it doesn't matter that much. Just think of it's allolactose is the ligand in this case. Lactose also works, but lactose is metabolized quicker. And there's other inducers that we'll talk about in a minute, some that are cool for molecular biology. So does that whole scenario make sense? So again, each one responds to their own signal. So CAP responds to glucose, repressor responds to lactose. And then the interplay of those two repressors and activators depends on if that gene's on or off. So it's a really, really handy system for the cell to know exactly when to have it on and off. So the way that they found out about the lac operon, how they did all this back in 1970 before we had all the tools that we have now, is they had to use mutants, and we still do this when we look at different things in the cell. If you're trying to elucidate what's going on with something, you basically break it, and then you see what you broke. So here's 3 of the mutants that they isolated that kind of led them to discovering what they discovered. So they found that you have this operator mutant. So the operator is the DNA sequence that binds the lac repressor in this case. So if you mutate that operator that binds the repressor, what do you think what's going to happen? Yeah, can your repressor ever bind? Let's say you totally just obliterate that sequence and it can't be recognized by the repressor anymore. If there's no repressor bound, what happens to your lac operon? It's probably going to look like this, right? Unless you have lots of lactose and no glucose, right? But you're basically not going to get repression, right? So your lac operon is basically going to be constitutively active because it's never going to be able to be repressed, right? So this operator mutant, so it's called operator constitutive because it's constitutively active because there's no repression ever, because the repressor just can't bind. So they discovered that mutant and found out that it found that what was mutated was that DNA sequence bound by the operator, or bound by the repressor. This one, the inhibitor minus. So what they did here is they found another mutant that basically it mirrored this one, so it looked like there was never any repression. But what they found was that they had a mutation in the repressor itself. So the repressor's DNA binding domain was not able to recognize the DNA. So you could see then if you can't bind the DNA with the repressor, then you're not going to get repression, right? So if you had this colony of cells and this colony of cells and you looked at them and you looked at them metabolically and you saw how they worked and you saw how they operated under all these different conditions, would you be able to tell the difference between these two mutants? Yeah, they would look the same to you, right? Neither of them can repress, but neither of them can repress for different reasons, right? This one's got a repressor mutation in the protein itself, and this one's got an operator mutation. So the repressor is fine, but the repressor just can't recognize the DNA because the DNA is broken. Yeah.
Are the sequences for the inhibitor protein like near
Yeah.
this, and that's how they could?
It's very, it's not, right, this shows it right next to it. It's like a few hundred down, or up, I guess.
Okay, so
100 up.
that's the, okay.
Yeah, so this is the last high, this is the
All of them
repressor.
or repressor.
Yeah, so this guy is made from this gene.
Thank you.
Yeah. And then the third mutant. So the third mutant is called super repressor. So the repressor can't bind lactose, thus it cannot be released from the operator site. So I can't remember what that one is. Okay, so this one's also a mutant that you basically, you can never turn on, so it's constitutively off. So you can never turn the repression off, so it's always repressed. So what would cause it to be repressed all the time? What do you think? So what causes repression to be relieved?
The binding of the allolactose.
Yeah, say that again.
It's missing the site for it to bind, so the spot where it would have it coming in.
To bind what? Yeah, to bind what?
To bind the ligand.
Yeah, which is allolactose, yeah. So it's super repressed, meaning that you can't turn it off because your repressor, it binds everything fine, it does what it should, but that repressor can't recognize its ligand. It's got a mutation in some site that allows it to not recognize the ligand. So if you can't recognize your ligand, you're never going to be able to relieve repression, right? Do you think this one you could fix somehow? So this is the one that you've got a repressor that can't bind DNA. So it's always off, right? Is there a way that you guys can fix it, do you think?
What you mean it's always on?
So this one's the one that's always off. Or it's always on, sorry. It's always, you can't repress, so it's always on.
Yeah.
Have you guys talked about like recombinant protein expression? Like if you have a plasmid in there expressing the protein, like expression in trans? That sound familiar? Okay, then we're not going to worry about it. But you could add a different gene to the cell that has a working repressor protein and express it somewhere else, and then that repressor could come and alleviate it. And that's the way you could tell the difference between these two mutants. Because that repressor protein wouldn't be able to alleviate this, but it would be able to alleviate that, right? Because the DNA is still broken in this case, where it binds. But that doesn't matter. So just worry about these being, I just worry about like, the consequences of these. And these are the kinds of questions that you would get. What happens if you have an operator mutation? What happens to repression under high glucose, low lactose, that kind of thing? Something like this. So how much beta-gal, so that's that first gene, so just think of it, how much of the lac operon is going to be expressed, high, leaky, or none? If you have a CAP operator mutant, so CAP can't bind the DNA, so CAP can no longer recognize under the following conditions. So if we have lots of glucose and no lactose, how much expression are we going to get in this mutant?
And how much lactose?
No lactose. So no lactose is added. So are you going to get any expression? No, right? Why? Yeah, the repressor's still working, right? So this mutant has no effect on repression, right? So if there's no lactose, you should always get repression, right? So it should be fine. So you should have it off, right? Would you say off or leaky? Off, right? You've got the repressor, you're off. How about plus glucose and plus lactose? Leaky.
No.
So do we have any repressor?
Yeah, we have repressor.
We don't have repressor, right? Because we have no lactose, or we have lactose now. So lactose bound the repressor's gone. We have glucose though, so do we want to make lac operon? No, we want to use the glucose up, right? So if we have no glucose, do we have activated CAP? Is CAP bound to CAMP? No, right? So you're going to have, we know it can't be high. Could it be none? It would probably be leaky, right? Because it's going to be this case down here. Nothing is bound at all. Aren't these fun? So again, no glucose. So this is good, right, for the cell? Well, not good for the cell, but it's good for the lac operon, right? No glucose means high levels of what?
Cyclic AMP.
Cyclic AMP, which means CAP is going to be activated, right? But what can't CAP do in this mutant?
It can't.
Bind DNA.
You can't bind, so if you can't bind, you cannot get activation, right? You have lactose, so there's no repression.
So leaky.
Leaky again. So
Yeah.
both of these are leaky. And then this one we just said kind of like this, right? If there's no lactose, you've got repression, so it's off, right? So those are the weird questions that you'll get. And you've got to understand this thing pretty well to be able to answer something like that, right? He might ask you something straightforward like this, which if he does, then you're good to go, right? But, or he might just not ask anything. I don't know. Your book doesn't really pay much attention to it. All right. So the last couple things probably will be just tools, how the lac operon can be exploited. So here's a kind of a confusing diagram, but it's not that bad. Showing how you can use the lac operon in molecular biology to express proteins. So let's say you have a protein from Sulfolobus that you want to purify and study, and you can't get it to express in Sulfolobus because Sulfolobus is hard to grow and it's a pain in the butt. Or you can't get enough of it or something. So you want to trick E. coli into making tons and tons and tons of your protein. So this is a system that you can use to try to accomplish that. So the way it works is you've got your E. coli genome, which is this mess here. You've got the LAC-I gene, which is your repressor, right? And then you have this thing here called a DE3. which is basically a polymerase, it's the T7 polymerase, so a polymerase gene under the control of the LAC promoter. So you need this T7 polymerase. So let's say that's one thing that you've got in your cell. The other thing you've got in your cell is this plasmid here. So you've got a plasmid P, which stands for plasmid ET, I have no idea, extraterrestrial, whatever. So it's the PET plasmid, which also has its own LAC gene, so you got more repressor coming from there. And it also has up here this sequence. So you've got your target gene, let's say that's the gene from sulfolobus that I want to overexpress in E. coli, fused to this T7 polymerase promoter. So this promoter is not going to be recognized by the E. coli RNA polymerase. So the only way that expression of your gene is going to happen is if you have the T7 RNA polymerase present, right? And cells typically don't have a T7 RNA polymerase, right? Unless they're infected by a virus, right?
What is the T7 RNA polymerase?
What's that?
What is the T7?
It's just the RNA polymerase that comes from a virus. So it recognizes its own set of promoters. And the reason they use T7 RNA polymerase in these things is because it binds really, really well. It binds really efficiently and you get lots and lots of transcription. So it's awesome at doing transcription. Much, much better than the E. coli polymerase.
What virus is it from?
T7. Phage T7, yeah. It's just called Phage T7. Yeah. They're really good at naming them. You have Phage T4. Yeah. So does that make sense? So that's our system. We've got our gene under the control of the T7 promoter, which also has a LAC repressor site in it, so it's repressed. And then we need the T7 gene in the first place because we need the T7 polymerase, right? So there's two different genes that we need to get expressed to get our gene, I guess is the way to think of it. So in order to start getting my gene made, I've got to start making T7 RNA polymerase. So in order to do that, you've got to basically remove LAC repression. How do we remove LAC repression? What do we have to add to the cells? Allolactose. But allolactose is metabolized by the cell. So if you add some allolactose, you'll get repression for a little while, but then the cell will start to break it down and then the repressor will bind again, right? So what really clever people, I don't know how they do this, they know the structure, but I couldn't do it. Let's just say that. So they found an inducer or something that removes repression but can't be metabolized by the cell. So it mimics allolactose. So here's allolactose. This is the inducer called IPTG, so we add that instead of allolactose to relieve repression. This can't be broken down by the cell basically because of this little sulfur bond. The enzyme can't work on it. This can be broken down, and then here's lactose just to show a comparison. So again, these two look really, really similar. It's just this bond here that is connected differently in different spots. So if you add IPTG, you just have to add it once, and then the concentration of IPTG is going to stay constant because the cell can't get rid of it. There's no enzyme in the cell that can break it down. So as long as you have IPTG present, you get no repression. So so you basically grow your cells up to a certain point. They're repressed all the way until you get them to a certain point. You add IPTG into your cell, and then what's going to happen? Yeah, you have a question.
Sorry, so it can't metabolize that because of the sulfur bond?
Yeah, I mean, that's not that important, but it's just, it can't cleave this. And if it could cleave this, then this wouldn't fit, and it wouldn't cause the alleviation of repression. So it sticks to the repressor, and then it basically can't come unstuck. Yeah. Okay, so you've got IPTG now added, so you've released repression. So once this guy bounces off, you get expression of this, right? The T7 RNA polymerase gene. It gets translated into the actual T7 RNA polymerase, and then your T7 RNA polymerase can go over to your plasmid, recognize its own promoter, and start cranking out tons and tons and tons of copies of your gene. Okay. Pretty cool, right? And don't worry about this. So you can inhibit the T7 RNA polymerase. Maybe don't even worry about this at all, but I think it's cool. And it's a good way to show that we use the LAC operon in molecular biology. It's not just to mess with you with questions like this. It's actually a useful tool. I think the main take-home is that you have other things that can alleviate repression, and those things can be useful tools like IPTG, things that can't get necessarily broken down. Skip that. So another way that the cell, so now we're out of operons for the time being. We'll come back probably. Do we have time? 10 minutes. So another way that the cell can mess with its own transcription, can regulate transcription, is through the use of alternative signaling factors. So remember, RNA polymerase is made up of several subunits. RNA polymerase by itself can't bind to the promoter, right? It needs the help of what's called the sigma factor, right? The sigma factor is the things that's responsible for recognizing the -35 and the -10 elements, right? So the cell has a whole complement of sigma factors. Most promoters are under the control of a single sigma factor called like the housekeeping like sigma 70 in E. coli. So there's usually just one sigma factor that is controlling most genes. So if the gene has the promoter recognized by that sigma factor, that gene will get expressed, right? If your gene has a promoter for a different sigma factor, it's not going to get expressed unless you have that sigma factor present, right? So the cell is able to basically pick and choose when it needs to express different sigma factors. So there's some called heat shock proteins. So heat shock proteins are proteins that get expressed in response to the cell getting exposed to heat. So things that will help it survive that heat blast for however long it might be. So those genes you're not going to want on all the time, right? Because normally the cell is going to be growing at 37 degrees, right? 37 C. But once you put it into a water bath or something in the lab, you start freaking the cell out, and the cell's like, oh, we're going to die. We got to start making these extra proteins that'll help us cope with this heat. So that's going to trigger the expression of an alternative sigma factor. That alternative sigma factor binds to your RNA polymerase, and your RNA polymerase can then start recognizing those heat shock promoters and start expressing those genes. And then once you take it out of the hot water, those genes get turned off, right? Because the heat shock promoters don't need to be used. You don't have that sigma factor around anymore. And there's other stress factors that you can use, not just heat shock, like UV radiation is another one. Okay, so that's alternative sigmas. Here's a way that a virus uses an alternative sigma factor to kind of another way of just being devious and messing with the cell. Yeah.
So once the, let's say the heat stress disappears, are the heat shock proteins just remodified or just degraded?
They're degraded, yeah. As far as I know. Who knows? It could be something else. I would imagine they're degraded. Okay, so here's a virus that basically has early genes, middle genes, and late genes, right? So early genes are going to help with replication. Middle genes are going to help with, they're going to start expressing like the proteins that you need to construct a virus. And then late genes are going to be the genes that you need to basically explode out of the cell typically. So in order to express your early genes, so this is when the virus first gets its genome into the cell and it wants to start its life cycle. So it's going to have to use the host RNA polymerase with the host sigma factor. So it's going to have a promoter that's recognized by host sigma factor, right? Let's say sigma 70. So the housekeeping polymerase hops on here, thinks it's just a normal gene, doesn't have any idea that it's a viral gene because it's just a dumb cell, right? It just does whatever's there. It's not a dumb cell. So it's going to express it, and it's going to express this gene, this gene, this gene, then this gene 28. Awesome, they just number the genes, which is an alternative sigma factor. So you need this alternative sigma factor to recognize the next set of promoters in the virus. So the middle genes are under control of a different promoter. So that sigma factor then kicks off this one. RNA polymerase binds to it. It's able to recognize this promoter, express, another alternative sigma factor. Same thing, kicks it off, binds the RNA polymerase, express, and by that time you've got your virus made. So the question here is, why don't you just use this promoter? Why even bother with these alternative sigma factors? E. coli, this one clearly works, right? You can use the E. coli polymerase and the E. coli's normal sigma factor on this one, so why don't you use it here and here?
Well, isn't it the lifetime of the
Yeah,
virus?
you
If
mean
don't
like-
read it all at one time.
Yeah, exactly. You don't want to make everything at one time. So this is a really clever way for the virus to just basically pick and choose when it needs to make its genes. Once you've got all your early genes made, at the end you get the sigma factor, which triggers the next set of genes. You get all those made, all of your assembly is done, you basically got your viral particles. Once you have your constructed viral particle by the end of your middle gene expression, you're ready to get out of the cell, right? You don't want the cells to die too early. You want to have the cell alive so you can construct a particle, right? If you don't have a particle and the cell dies, then there's no point, right? The virus isn't going to be able to go on, right? Does that make sense? So then at the end of your middle set of genes, you get another sigma factor that triggers the set of final gene expression. Those genes are going to be what goes and causes the cell to die, and then the cell death releases your completed virus. So another devious way. Any questions on that?
Generally speaking, are bacterial promoters all strong?
No.
So they too have their own variants.
Yeah, she asked if bacterial promoters are all strong. You mean virus promoters or bacterial?
I guess you could, I don't know.
Yeah, so I mean, they're not all strong and they're not all weak. It's just another control mechanism. So like the lac operon has a weak promoter, and it's got a weak promoter, which helps it regulate. I mean, it's another level of control and expression of that lac operon. But no, I mean, you can't, yeah, they're not all strong and they're not all weak. There's just kind of a mix.
Is the strength or weakness of a promoter mainly due to how close it is to the consensus sequence?
Yeah, that's part of it. And then we'll see, I think I have an example of, yeah. So this is an example of not only the consensus sequence, but the distance between those consensus sequences. So how far apart those times 35 and times 10 might be. So this is a really cool example. It's got basically perfect minus 35 and minus 10 elements. It should recognize the E. coli RNA polymerase sigma factor, no problem. It should just hop right on it, it should express, but it doesn't for some reason. And the reason is because the distance between these two is wrong. So it should be about 15 to 17 base pairs, and in this case it's too long, it's 19 base pairs. And you guys know that DNA is a double helix, right? So it twists around. So the minus 35 and the minus 10, they should both be like in this picture. They should both be facing up so that the RNA polymerase sigma factor can hop on right there. It should be able to recognize both of them. They're not in this case, they're in the right position, but in this case, one's facing down and one's facing up. So RNA polymerase just can't. It has the right sequence, but it can't recognize it really well because it's just, the orientation's wrong, and that's just due to DNA conformation. So this repressor here is actually a repressor activator, kind of. It does both things. So again, common theme. And it does it in a different way. So this guy's not going to make any contact with RNA polymerase at all. The way that it works is strictly through DNA contacts. So in this case, it binds its operator, which is, again, between the minus 35 and the minus 10. So in this case, it's repressing. So it's keeping this DNA locked in this position. This DNA, you know, flops around in the cell, and it can twist on its own. So normally it would be able to twist back into the right place and back out and right back in. But this guy keeps it twisted out of place. So RNA polymerase just can't bind. It just keeps it in that wrong, incorrect conformation. The binding of the ligand, in this case, which is mercury, causes a conformational change in this repressor, and it causes that conformational change, which causes the DNA to then twist. And it locks it now in that twisted conformation. By twisting the DNA, you've positioned both of your minus 35 and your minus 10 elements in the right spot, and then RNA polymerase can come in and bind and express the genes. And this is this one's in response to mercury as like a toxin. It's going to help the cell get rid of mercury. Yeah.
Would that be considered supercoiling in that section of the DNA? Or just
Twist. Yeah. Good question. Yeah.
So in the top one right there, where they're in the wrong position, so not
Yeah.
only is the gap too big, but minus 10 is in the wrong
Yeah, so the reason that minus 10 is in the wrong conformation is because that gap is too big.
Okay.
If you shorten it, right? So as you go out, this thing starts to twist around and around and around, right? It's going to face a different side. So if you move them closer together, you can kind of see how it would twist back up into the right spot, right?
Thank you.
Yeah. So the take home here is MER-R, the thing that's repressing and activating. It has no contacts with the polymerase at all. It's doing it completely through the DNA, which is different than what we've seen. But it's also repressing and activating, and it represses or activates depending on the availability of that ligand, right? Cell gets dumped into a mercury bath, which is not good for any kind of cell, right? And become crazy unless it gets rid of the mercury. So mercury triggers this guy to change shape. These genes probably encode some kind of pump that can then pump out the mercury. That's my guess. Who knows? Okay, so that's it.